This video should provide you with information how to conduct the lab simulation. Once you log into the course, you click on the course document, and once you're in the document page, scroll down, go to the lab reports, and it's first lab, so you're gonna click right over here for the first lab. Once you're in this page, the first thing you're gonna do is you're gonna watch this video how to perform, and this is the instruction how to conduct the lab, and this is basically information how your lab should look like. Once you looked at all of them, this, like, you just scroll down, and this is the lab simulation right over here. And now suppose this is a cell, and suppose this is another cell, and now suppose this one is a membrane, and I'm taking this membrane and putting it in between the two cells. The whole purpose over here is that we're looking how molecules could go across the plasma membrane. So we're looking at diffusion, osmosis, active transportation, and so forth. Now in order to look at that one, I have to fill up this with some kind of solution, and this one with some kind of solution. Now suppose I put over here 6 molar of the sodium chloride, and I put no urea, no albumin, and no glucose, and I'll just fill up this one, and over here I will just put water, and then I will start the simulation. And as the simulation runs, what would you expect to happen? We have a lot of sodium chloride here, so you would expect sodium or chloride to cross to this side, and you would expect some water from here to cross to the other side. So as the simulation runs, you're going to see it over here, and that's how you're going to write your lab report. And at the end, it tells you over here that sodium chloride did not diffuse in this case. You record your data, and now I will flash this, both of them. Now let's do something else. I'm going to add aluminum, I'm going to add glucose, I'm going to add urea, and this one just deionized water, and I will dispense this, I'll dispense this, and again you run the simulation and you just look for the results. I'm not going to wait too long for the entire duration of the simulation, but let me pause it and give you an idea. So over here, urea, no diffusion, no diffusion, no diffusion. What else you could do? You could just change the membrane, and you could get a different membrane now. Now each membrane over here, you see, has 20, 50, 100, 200, and when you read the instruction, you will understand what those means. And you're going to conduct the lab with this one. At the end, you could go to the experiment, and you could do facilitate the diffusion. You could go osmosis, you could do filtration, same exact thing. You're going to just take the membrane, you're going to put it right over here, you dispense this with some kind of water, and you're going to say how much of that water is going to be filtered here. The whole purpose of this lab, when you're writing your lab report, is that you should indicate why, for instance, like sodium chloride couldn't diffuse, why glucose could diffuse in one case and couldn't diffuse in the other case. So the whole purpose is that you're doing the experiment, and then you have to discuss it, why something happened and why something did not happen. Hope this helps you understand how to do the lab.